90 kilo base pairs, that is all it took. For decades, scientists agreed that no cell could survive with a genome smaller than 113 kilo base pairs. That number was considered a hard floor. Go below it, and the cell simply would not have enough instructions to stay alive. SpudCell has a genome of 90 kilo base pairs, and it is alive, or at least it does everything alive things do. Researchers at the University of Minnesota built it entirely from non-living chemicals. No bacteria were modified, no existing cell was hollowed out and refilled. The team assembled the chemistry from scratch, and the result takes in nutrients, grows, copies its own DNA, and divides into daughter cells. The first fully synthetic cell to complete an entire life cycle. Here is the part that should stop you cold. Reproduction is the defining feature of life. It is what separates a crystal growing in a lab from a bacterium multiplying in a petri dish. SpudCell reproduces, which means, by the most fundamental definition biology has, it is completing a life cycle without ever having been alive. For centuries, the line between chemistry and life was considered uncrossable. SpudCell crossed it. The project was co-led by Kate Adamala, a synthetic biologist and associate professor at the University of Minnesota. The cell is called SpudCell, a deliberately unassuming name for something this significant. Previous attempts at synthetic cells could mimic parts of cellular behavior, a membrane here, a chemical reaction there. SpudCell does all of it in sequence from a genome that biology said was too small to work. Think of it like building a functioning car with 20% fewer parts than any engineer thought was possible. The car still drives, you just do not know why yet. That why is now one of the most open questions in biology. The science world is not entirely convinced, and that is worth understanding. The research has not been formally published in a peer-reviewed journal. It was shared through Biotic, a new non-profit bioengineering institution that Adamala helped found. One reviewer at Cell, one of the most respected biology journals in the world, reportedly told Science magazine that the project was not real biology. Adamala's response was direct. She stated, "We've replicated in chemistry what only used to be possible in biology, the complete set of behaviors of a cell. It proves that the most fundamental functions of life, like growth and replication, do not need a mysterious magical spark." That phrase, mysterious magical spark, is a direct challenge to the old idea that life requires some special ingredient beyond chemistry. If peer review validates the findings, it would mean life is a process, not a substance. That chemistry arranged correctly becomes life. The applications, if the science scales, are significant. Synthetic cells could be engineered to produce medicines inside the body, targeting specific tissues. They could break down pollutants or manufacture biological materials that are currently expensive to produce. In space biology, synthetic cells could theoretically support long-duration missions by producing food, medicine, or oxygen from basic chemical inputs. None of that is imminent. SpudCell is a proof of concept, not a product. But proof of concept is where every revolution starts. Subscribe if a story like this is what you came here for. SpudCell's genome is 90 kilo base pairs, 20% below the number biology said was the minimum, and it reproduced anyway.